Non-covalent hydrophilic immobilization. Immobilization is an important step in the fabrication of a biosensor. There are two types of immobilization. One is covalent immobilization and the other is non-covalent immobilization. The non-covalent immobilization is the method of choice because in this case we are sure that most of the biological activity or the binding receptor binding activity of the molecule will remain intact. In case of non-covalent immobilization, there are main two types of non-covalent immobilization. Number one, we use different types of weak forces, or the other is we can use different types of structures which can engage the molecule. पिछले मॉड्यूल्स में हम इन स्ट्रक्चर्स के मुतालिक डिटेल से पढ़ चुकी हैं. डायग्नोस्टिक बायोसेंसर के रेफरेंस से I will talk about the non-covalent hydrophilic and hydrophobic interactions. So we will study this topic in detail and we will cover the types of forces which are involved in the non-covalent interaction, the amino acid which is the basic component of a protein because we use protein as enzyme or an antibody in the as a bioreceptor element. So we will study one of them, the amino acids. Then we will study the hydrophobic amino acids and the electrostatic binding of proteins. We will study the electrostatic binding and structure of DNA molecule, and we will summarize the topic and then we will discuss the related question. So the first thing is that what type of forces that are involved in the non-covalent interaction. These are weak forces, for example, hydrogen bonding, hydrophobic interactions, van der Waals forces, different types of affinity binding, and the mechanical confinement. We had discussed this topic in our previous module. Because we are dealing with the protein molecules which act as our bioreceptor element, as enzyme or as antibody. So we will study the structure of, revise the structure of the amino acid. Amino acids are the monomeric building blocks of the protein. The alpha carbon of the amino acid which is adjacent to the carbonyl group is attached with the four important groups. The one is the amino group which is shown in here. The nitrogen is shown in green. The carboxyl group and the one hydrogen atom and one variable group. The chemistry of the amino acids and the protein is dependent on this variable group. This group determines whether the amino acid will be hydrophobic or hydrophilic, and all these amino acids which are present in the primary sequence of the protein determines the global chemistry of the protein molecule. So we will study the hydrophilic amino acids. The hydrophilic amino acids are those amino acids which are charged. Either they are protonated or deprotonated. They are charged in their both forms. They could be acidic, basic, or polar. So in this slide, you can see the structure of hydrophilic amino acid. On the in pink color, we can see the tail, and this is the tail of the R group, or the R group that is responsible for the functioning of the determining the hydrophobicity or the hydrophilicity of the molecule. So these groups which I have encircled, the side chain which I have encircled, are very important for the hydrophilic character. The amino acids that are included in hydrophilic amino acids are lysine, arginine, histidine, aspartate and glutamate, serine, asparagine, glutamine and threonine. All these amino acids are considered as hydrophilic amino acids because they are reside happily in water. Now, what about the global chemistry of the molecule? यानी कि molecule की global chemistry पे क्या effect होगा in amino acids का? यानी कि जितने ज़्यादा hydrophilic amino acid होंगे तो उतना ही ज़्यादा hydrophilic character होगा, but still we have to discuss it. So the main thing which we are going to discuss is charge on the protein. Charge on the protein there is a specific point which is known as isoelectric point. Isoelectric point is a point where protein has no net charge. How it could be possible? हमने अभी देखा कि उसमें इतने charge groups मौजूद हैं. The thing is the number of the positive charge is equal to the number of negative charge, and as a result there is the net charge is 0. At this point the protein is highly insoluble, but below and above this charge above this point protein can get a charge. It can acquire a charge above or below the isoelectric point, and this it means that we can control the charge on the protein by selecting a particular pH of the protein molecule. So we will discuss an example. I'll give you an example of lactase enzyme. Lactase enzyme is an enzyme that is used for the biodegradation of lignin. It requires copper for its activation. And what about the isoelectric point? It has an isoelectric point of 3.3. It means that at pH 3.3 this protein is highly insoluble and has no charge, but below and above the pH 3.3 what we can do is we can charge the molecule. So we can control the global chemistry of the protein. And if we use a buffer in which the protein is positively charged, as you can see in here, this positively charged protein can bind with the negatively charged support, and the support what we are talking about is obviously a transducer. The layer of the transducer is negatively charged, so the positively charged protein can attach to the negatively charged surface of the transducer. This is how we can use the electrostatic binding. Now we can also use the electrostatic binding approach for DNA molecule. We know the DNA molecule is negatively charged because DNA molecules has got phosphate groups, and these phosphate groups are hanging out and are negatively charged. Basically DNA is composed of basically composed of building units which are known as nucleotides. Each nucleotide is composed of a phosphate group, a sugar, and one of the 4 types of any bases in GATC. Now if you look at the structure of the DNA molecule, so what we are interested in this phosphate group, and this phosphate group is hanging out and there is a charge on it, and there is an alternate sugar and alternate phosphate, alternate sugar and alternate phosphate. So the DNA molecule is negatively charged, and these negatively charged groups are available for the binding. Now you can see the surface of how we can use the DNA in the transduction process. There is a modified electrode, and the surface of the electrode has got positively charged, and this is the DNA molecule which we are going to use as a probe, and it has got negatively charged, and this positively charged group can be immobilized on the surface of the modified electrode, and on top of it we will still have the phosphate group which can negatively charged phosphate group which can react with our bioreceptor element. So what we have learned so far is that the weak physical forces are involved in the hydrophilic immobilization. We can use these forces and can immobilize DNA molecule and protein molecule on a charged surface. Proteins and DNA molecules could be attached on the modified surface that are oppositely charged to the charge on the DNA or the protein molecule. Now the related questions: what are the forces that are involved in the non-covalent immobilization, and how we can immobilize the DNA molecule by using electrostatic charge, and name the factors that determine the hydrophobic or hydrophilic character of the molecule.